Hello, my name is Marc de Meyer and I'm an entomologist specialized in the identification, taxonomy and systematics of African flies, in particular Tephritidae fruit flies. I work at the Royal Museum for Central Africa, which is based in Teveur, Belgium. This presentation deals with a general introduction to the fruit flies. It is part of a series developed within the framework of the FFF Fruit Fly Free project, supported by the Standards and Trade Development Facility. In this presentation, I will provide some general background on the family, give its taxonomic position compared to other flies, provide diagnostic characters to differentiate representatives of the family from other closely related groups, and discuss the general biology of the group. Fruit flies are referred to as the family Tephritidae. It is one of the more specious groups of flies, comprising about 5,000 different species grouped in 500 genera. The sub-Saharan Africa has more than 1,000 species and about 160 genera. Fruit flies are considered a group of main economic significance because a large proportion, about 40%, are known to infest fruits in their larval stage. Several of them infest fruits that are grown as crops and are as such considered pest species. A limited number of fruit flies are, however, also considered as beneficial insects because they attack weeds and can be used as biocontrol agents. We will come back to the biology further in this presentation. Within the insects, fruit flies belong to the order of the Diptera or two-winged insects. All flies are characterized by the presence of a single pair of wings. Most other insects have two pairs of wings. Only the forewings are fully developed and usually transparent. They show a network of lines called veins and cross veins, and most fruit flies also have darker markings or patterns. The hind wings are reduced to small knobs which are called dihalteres. On this picture you see the position of the dihalteres on the body of a fruit fly, and on the right hand side an enlargement of the structure encircled in red. The order of the Diptera is further subdivided into suborders. The suborder Nematocera are the mosquitoes and midges. The suborder Brachycera are the more so-called primitive flies, such as rubber flies or horseflies. Fruit flies, or Tephritidae, belong to the suborder Cyclorapha. This suborder is divided into superfamilies, which in turn are divided into families. The family Tephritidae belongs to the superfamily Tephritoidea. The family Tephritidae itself is then divided into genera. The most common ones of economic significance are Bactrocera, Cerathitis, Dacus, and Zogodacus. This will be presented in more detail in a separate presentation. Each genus comprises a number of species. In addition to Tephritidae, the superfamily Tephritoidea comprises several other fly families, such as the Longiidae, Platystomatidae, Pyrgotidae, and Eulididae, of which you see here a number of representatives. Several of them resemble closely fruit flies with similar marked wings and can therefore be easily confused with the true fruit flies. The main diagnostic character to differentiate the fly of the fruit fly family from other flies is the shape of the subcostal vein or vein SC in the wing. The wing of a fruit fly is characterized by a pattern of longitudinal veins, transversal cross veins and cells. This is explained in the presentation on morphology, and I refer you to the latter for further details. The subcostal vein is present in the basal part of the anterior wing margin. Its position is marked on the image at the bottom. It is a small vein which runs in parallel to the costal vein that is demarcating the anterior wing margin. In all other flies, the subcostal vein is gradually sloping towards the anterior margin and ultimately merges with the costal vein at the margin, as shown in the figure at the top left. However, in fruit flies, the subcostal vein keeps running in parallel and then suddenly, at the distal end, makes a sharp angle of about 90 degrees in order to end up perpendicular to the costal vein, as shown in the figure at the top right. Often, this perpendicular part is not well developed and only present as a faint mark. In the following slide, you see the position and shape of the subcostal vein in an image of the wing of some typical fruit flies, in particular of a Tephritidae species and of a fruit fly representative of a day sign, referring to the genera Bactrocera, Dacus and Zogodacus. At the bottom, the approximate area is marked with a red rectangular box. This box is then enlarged above the general figure and the distal part of the subcostal vein, running perpendicular to the anterior margin, is encircled in red. Another characteristic that is shown in the fruit flies and absent in the vast majority of other flies within the Tephritidae is the presence of inclined frontal bristles. Frontal bristles are usually present as 2 to 3 pairs. In some groups like Anastrophe there can be several more. The frontal bristles are present on the front of the head, which is the area between the eyes and above the antennae. A more detailed presentation of these bristles and how to differentiate them from other ones on the head is given in the presentation morphology. The position of the frontal bristles is indicated by the red arrow on the figure at the left. In related families of the Tephritidae, the frontal bristles are absent and only smaller hairs can be found, as shown in the picture on the right. In this slide, we see a frontal view of the head of a typical Tephritidae at the left and a typical representative of a daysine fly on the right-hand side. The frontal bristles are indicated by red arrows. Remark that in some groups, like daysines on the right, the frontal bristles may be short and less strongly developed. Female fruit flies also have a characteristic feature at the end of their abdomen, an ovipositor. The ovipositor in fruit flies is a fully sclerotized conical structure at the distal end of the abdomen. It encapsulates the actual piercer called the aculeus, which is a needle-like structure. This structure is retractable and can be withdrawn into the ovipositor. The aculeus is very hard and sturdy enough to penetrate the skin of a host fruit, so that the female can oviposit her eggs inside the fruits. In other related fruit flies within the Tephritidae, one can sometimes also see an extended structure at the end of the abdomen. But here it is usually less sclerotized and more membranous. It can also not be retracted completely inside the ovipositor. In these groups, the structure is mainly used for depositing eggs on the surface of rotting vegetable matter, but not to penetrate the skin of the fruit. An example of such an ovipositor is shown at the right. Finally, there is the general appearance of fruit flies in having patterned wings, either with bands or with spots, but their bodies usually not being metallic in appearance. However, These characters are not sufficient to differentiate true fruit flies from close relative groups, so should only be used as a general guideline. The earlier listed characters are more reliable. The vernacular term fruit flies is causing some confusion, as the name is also used for other families of flies, namely the Drosophilidae. Both Tephritidae and Drosophilidae are associated with fruits, but are not related to each other. Drosophilids, are also known as vinegar flies, are often encountered in-house. They are smaller flies that will usually be found hovering above or sitting on rotten or overripe fruits. They feed on microorganisms that can be found on these substrates but will not attack fresh, undamaged fruits. There is, however, one exception. The wing-spotted Drosophilid, Drosophila suzuki, is an actual pest of fresh fruits. The fly is of Asian origin, but has in recent decades been expanding its geographic range. It is considered an invasive species, first in North America, but in recent years also in more and more European countries. Very recently, it has also been reported from the African continent, in particular from Kenya and from the Comoro Islands in the Western Indian Ocean. The larvae feed, just like those of Tephritids, on the fruit tissue of undamaged fruits. Rather than piercing the fruit to deposit her eggs, the female will scratch the outer surface of the fruit with an ovipositor that is shaped like a grater. The larvae, once hatched, will then enter the fruit. The wing-spotted Drosophilid feeds mainly on soft fruits such as berries and cherries. Tephritiid fruit flies have different strategies regarding the development of the larvae. Although called fruit flies, not all Tephritids are fruit feeders. Depending on the larval feeding behavior, we can recognize 4 different groups: saprophages, leaf miners, flowerhead feeders, and fruit feeders. A few flies in the family are saprophages. They feed on dead and partially decomposed vegetable material such as rotten wood. Representatives of this group are mainly found in Asia and Oceania. The most famous ones are called the antler flies. The name refers to outgrowths on the cheeks in the male, which are resembling antlers found in deer or antelopes. The males develop a territory on rotting tree stumps, and they defend these by head-to-head collisions with competing males, similar to stag fights observed in deer. Females are then invited to lay their eggs in the rotten wood within the territory of the male. A second group are the leaf miners. The larvae can develop in leaves of their host, but also in the stems or roots of the plants. Some species are considered as pests because they are known to infest crops such as celery or asparagus, hence the names of celery fly and asparagus fly. The largest diversity is found in bamboo shoot flies or gastrozonines, found in Africa and Asia. Representatives of this group infest mainly grasses. In Asia, these are mainly bamboo species. In Africa, the host range is poorly known. Flowerhead feeders comprise about 40% of the total family diversity. Representatives of this group form galls in flowerheads and buds, where the larvae feed on the developed gall tissue. Contrary to fruit feeders, the larvae will pupate inside the flower head, and the adult fly emerges thus directly from the flower head. The majority of the known hosts belong to the plant family Asteraceae. As such, they may cause some damage to ornamentals. There is a group, however, that is used as biocontrol agents. This is referring to invasive weeds, such as thistles, accidentally introduced into new origins, but for which no active local predators are known. Fruit flies are then introduced into these regions as well. As they infest the flower heads, seed production is hampered, and thus the flies can contribute to the control of these weeds. The last group are the actual fruit feeders. They also comprise about 40% of the family. In general, they are better studied taxonomically than the other groups. Nevertheless, species new to science are still recognized as such. Fruit feeders can be specialized on a limited number of hosts, or can be very polyphagous, laying eggs on an enormous diversity of hosts. In both groups, several of these hosts are commercially grown, hence the fact that they are considered as pest species in this group. However, worldwide, the majority of these pest species belong to just 6 genera. These are presented in a separate webinar called Main Genera. The larvae feed on the fleshy part of the fruits, and in some cases also on the seeds. Contrary to the flowerhead feeders, the larvae, once full grown, leave the fruit and pupate in the ground, from where they will emerge as adult flies. Many thanks for your attention. This presentation was created in collaboration with Stellenbosch University. We would like to thank the funding agencies, as well as the individuals and other research groups who in one way or another contributed to the actual development of the presentation.